Hey, Brad, you all set there? Okay, good, thanks. Welcome, everybody. Sounds loud, but it's a pleasure to introduce Dr. Dan Meacham today from George Washington University. We've already had a couple visits from the same lab, so part of this story will be familiar to people who have been here before, but we'll hear specifically about the parts of this that Dan has been working on over the last few years. Dan is a senior research scientist at the GW Institute for Neuroscience at George Washington University in DC. He did his undergraduate work in biochemistry at Brunel University in London. He then went on and did a master's degree in molecular medicine at Imperial College in London. And from there, went on to do his Ph.D. in human molecular genetics at Sheffield. He then came to the United States and did postdoctoral work at UNC in Chapel Hill, and joined up with Anthony LaMancha and moved on to George Washington University as part of the Institute for Neuroscience there, where he's been for the last decade or so. He won, along the way in his training, a Brain and Behavior Research Award, an RSAT Award, a Young Investigator Award, which are very competitive, as many of you know. And he's been involved pretty much his whole scientific career with a focused approach to understanding development and the role of genes and development of the brain in particular. As a graduate student, he started out doing genetic and functional studies looking at leptin receptors and body weight. And then he went on during his postdoctoral period to focus primarily on neurodevelopmental disorders, taking a really multidisciplinary cellular, molecular, and behavioral approach to understanding those, and is focused very much over recent years on the 22 Q11 Deletion Syndrome, DeGeorge syndrome, and has made, I think, really exquisite use of this animal model over the last few years. It's a deletion syndrome model in the mouse that he's studied in vivo and in vitro. This is a copy number variation model, for those who aren't as familiar with it. Has a very strong genetic association with neurodevelopmental disorders and psychiatric symptoms, as well as related to somatic symptoms, which include cardiac, dysphagia, and other aspects of development. And so he's really focused on, I think we're going to hear about, I hope I'm not wrong, but I think we're going to hear about where he's focused down on a population of cells, particularly supragranular layer 2-3 pyramidal neurons in the brain, and how this deletion syndrome, particularly an individual gene, is involved with leading to changes in the morphological connectivity, both locally and at distal sites across the brain, and then focus that down on understanding mechanistically how mitochondria contribute to this process, and particularly the production of reactive oxygen species, and how we can actually not only demonstrate this single gene relationship, but rescue it, and again, a very elegant parallel series of both in vitro and in vivo studies that kind of coalesce to what I think is an important new view of how this syndrome works. So it's a real pleasure to have you here, Dan. Look forward to your talk.
All right, so so I'm interested in understanding cortical circuits that are very important for higher order cognitive functions. So when we think of that, we tend to think of things like abstract thought, executive processes, reasoning, and not only that, I'm interested in understanding if there perhaps is a genetic architecture that can actually underpin the cortical circuits that are required for these higher order processes. So in humans, it's known that there are these what's called cortical association areas that are critical for these to be able to perform these higher order functions. So this would be different from your somatosensory, sensory perception, or motor control areas of the cortex. So these occupy distinct areas in the human cortex, and then not only that, because they occupy these distinct areas, then you'd need to have interconnections between these regions to what is essentially a network state. Luckily for my work, there is some correlation in terms of areas in the mouse brain that can actually be related to what's called these cortical association areas in humans. And I've indicated those with asterisks. So you have these regions within the mouse brain and the human brain that are interconnected via white matter tracks. And then, so we can study that in the mouse, but not only that, we can also study more locally the microcircuits that are the key components at the local level that not only form interconnections, but also have a local microcircuit role. So this would be your projection neurons, I've highlighted in orange here, which essentially form these long-range connections within the cortical association areas. So they have the axon bundles that will come out and form the white matter tracks between these different regions. And then also the locally acting cortical inhibitory interneurons, which essentially have a local role in inhibiting the activity of those projection neurons. So you've got these two key classes of neurons that will form this microcircuit, and which then will have an interplay between these cortical areas that are important for these higher order cognitive functions. So as alluded to by Mike, if I'm interested in understanding the circuits that govern these behaviors, these association cortical areas, is there perhaps a genetic angle where we can look at this in terms of perhaps neurodevelopmental disorders which are known to affect these higher order cognitive functions, and therefore would be a way to study these circuits? Is there a particular genetic-based neurodevelopmental disorder by which I can actually achieve that? And I've personally studied for quite a while now a condition called 22q11 Deletion Syndrome. And this could be described as a model genetic neurodevelopmental syndrome. Essentially, it arises from a 1.5 megabase pair deletion on human chromosome 22. It's associated with a range of clinical features, many of which must arise embryonically because at birth there are already changes apparent. So, for example, there's subtle craniofacial structural changes, and also, probably most famously, there's heart defects that are associated with this. So happens quite nicely is that in mouse chromosome 16, there is a syntenic region that basically contains the same genes that are present in the deletion in humans. So I should just point out, if I didn't already, this is essentially a heterozygous deletion. So you're essentially missing one copy of this 1.5 of genes that are within this 1.5 megabase pair region in humans. So in mice on chromosome 16, it's been engineered, basically a replication of that mutation whereby you have the same genes heterozygously expressed. And this model is called the Large Deletion model that I've worked a lot on. And essentially that is what I've used to try and assess these cortical circuits that I mentioned. So I briefly alluded to this notion that there's effects that are already occurring embryonically that would be apparent from the human condition. So basically I'm trying to start from the initial assessment of what's going on embryonically during the development of the cortex. That will lead to the neurons that will basically form these cortical circuits. And so essentially we're interested in here is basically the genesis and then the movement of these newly born neurons that will go to form the microcircuits in the cortex. So what is the first point that I looked at in the past? Well, this was basically, I mentioned you have the interneurons and the projection neurons. So in the first instance, let's look at these cortical interneurons, these cells which will go on to have this local inhibitory activity in the microcircuit of the cortex. So I'll point out now that basically these cells are born in a structure that's not the cortex. It's a basal forebrain structure that's called the medial ganglionic eminence. I'll call it the MGE here for short. And basically these nascent cortical interneurons are born here and will move tangentially into the cortex. Okay, so in the first instance, I'm interested in looking at the genesis and movement of these cells. So then we'd want to look at the genesis of these cortical interneurons. So basically using markers of proliferation, which is BRDU, and also GFP here, which labels the nascent cortical interneurons born in that MGE structure. And when we did this kind of analysis, we essentially saw that there was no significant difference in the genesis of this particular cell class. However, when we looked at the migration of these cells, so coming from the MGE and into the cortex, we saw some interesting changes. So again, this GFP is labeling these nascent cortical interneurons moving into the cortex. And what we saw was during this developmental stage, there was actually a reduction in the initial wave of these interneurons moving into the cortex, and they were slightly more disorganized in their movement into the cortex. And that's shown there. And a whole range of experiments I did. I won't cover them all, but just basically give you a brief synopsis. I essentially showed that there was a particular protein, CXCR4, which is a receptor which is expressed on the surface of these nascent migrating interneurons. And essentially, we showed that this is diminished in expression and also phosphorylated activity in the large deletion. So this would be the large deletion embryonic cortex. So there's a CXCR4 decrement, and then we actually employed a genetic cross to actually exacerbate that effect. So down here, we actually have CXCR4, which is expressed heterozygously on a large deletion background, and that exacerbated this disorganized migration of cortical interneurons into the cortex. And then when we looked postnatally in the cortex, so this is, what is the outcome of these embryonic changes? I essentially labeled the mature cortex with a number of interneuron subtype-specific markers. What I saw specifically was a rearrangement of parvalbumin-positive cortical interneurons, where there was essentially the appropriate number of cells were present, but there was a shift in their laminar location. Okay, so that's the interneuron, and so I showed embryonically that there is a migration defect that leads to the rearrangement, essentially, of these cells in the mature cortex. So how about the other key component of the microcircuit, which would be the projection neuron? Well, these cells, unlike the cortical interneurons, are actually born locally within the cortex. There are two main pools of progenitor cells that reside in the embryonic cortex that give rise to projection neurons. These would be apical progenitors and basal progenitors. So again, employing this approach of looking at proliferation capacity of these progenitor pools in the embryonic cortex, I labeled again with BRDU, a proliferation marker, and then I co-labeled with TBR2, which is known to specifically label these basal progenitor pools, proliferative pools. What I saw was that there was actually a specific reduction in essentially the double-labeled cells, the TBR2, BRDU double-labeled cells in the large deletion versus wild type. I'll point out that I did not see a reduction in the apical progenitor pool, so that was seemingly spared, but not the case with the basal pool. So that basal progenitor pool is essentially considered to give rise predominantly to upper layer 2-3 projection neurons in the cortex. And when I used specific markers to look at upper layer projection neurons, there was a reduction in that particular cell class. So far I've mentioned to you this notion that there is a rearrangement of cortical interneurons, and that is seemingly combined with a reduction in upper layer 2-3 projection neurons. So what might be a consequence of that projection neuron observation? Well, one way we can go about that is by doing essentially stereotactic injections in the mature cortex, and then we can look at, as I said, these layer 2-3 projection neurons essentially have these long-range connections, these long-range axonal connections. So if you inject at a specific location, so here would be, I'm calling it the medial frontal anterior cortex. You could also call it the anterior cingulate. So this is one key area of the cortical association areas that I mentioned earlier. It's considered to be kind of an integrative site that integrates the communication between these cortical association areas. And when I inject at that point with biocitin, I can then essentially look at all the cell bodies, retrogradely labeled cell bodies from predominantly layer 2-3 projection neurons that project into that injection site.
And
yes?
Maybe you get to it, but is there any difference in ipsilateral cortical versus callosal contralateral cortical?
In the large deletion specifically, yes, I will get to that, and yes. So then, essentially, the methodology that we used was to basically plot the retrogradely labeled cell bodies from the projection neurons that project into that injection site. Predominantly, although there is somewhat of a range, but predominantly there is, when you look at the laminar distribution of the labeled cell bodies, it was predominantly more of an upper layer grouping of cells that projected into that MFAC region. So then when we looked at the wild type versus large deletion, did we see any specific changes? This is a slightly busy slide, but essentially we're comparing at anatomically comparable regions between the wild type and large deletion through the brain, through the cortex. And then we're just basically assessing the relative numbers of these retrogradely labeled projection neurons between the wild type and large deletion at equivalent sites. And essentially, for example, what we did see was that there was a reduction both ipsilaterally and contralaterally in the large deletions compared to the wild type when we looked at specific locations. So that would be the black bars, essentially. It's kind of a relative density, I guess, difference between the wild type and the large deletion. So yes, there are contralateral and ipsilateral reductions in the density of these projection neurons that project into the MFAC region. Although interestingly, more locally to the injection site, there in some instances there was perhaps exuberance of labeled cells in the large deletions. That might be suggesting some kind of spatial, perhaps, compensation. I shall point out, though, that one key observation was that this lateral entorhinal cortical region, which is another key component of the cortical association network, that was specifically reduced in terms of numbers of layer 2-free projection neurons in the large deletion. Okay, so bringing that together from what I've described to you so far, we have this interneuron redistribution. We have a reduction in the number of layer 2-3 projection neurons. One might assume that that would perhaps result in reduced numbers of long-range cortical projections because you have a reduced number of cell bodies of that particular cell class. And indeed, that appeared to be that is the case in the large deletion through our retrogradely labeled experiments. Okay, so that's looking at development, and then that's also looking at the long-range connections. Now I'm going to home in a little bit more on the morphology of those individual layer 2-3 cortical projection neurons in the large deletion model. And we essentially used a genetic labeling approach whereby we could sparsely label layer 2-3 projection neurons in this mouse line because it is CUX2 expressing line, which is basically a layer 2-3 specific marker. And then we can, it's basically a conditional approach where we can also temporarily regulate the expression of GFP in these cells via addition of tamoxifen. So it's a very nice approach because you can sparsely label these cells within the cortex. So if you want to do measurements of dendritic complexity, for example, you don't want to have a cortex that is overloaded with labeled cells. So what were the results of that genetic cross that we did onto the large deletion? Well, what we saw was that there was quite a significant reduction in the apical dendrite complexity in individual large deletion layer 2-3 projection neurons relative to wild-type counterparts. And that's quantified down there. So we not only do you see changes in dendritic length, we also see changes in reductions in branch point and branch complexity. That was actually specific to the apical dendrite and not so much in the basal dendritic structures. And then bringing it down into even a more high-resolution approach, looking at these layer 2-3 projection neurons. I took advantage of a wide field block face scanning EM approach, whereby you can image at low mag fairly large areas of the cortex, and then from that you can home in on specific, for example, in my case, neurons that are known to reside in specific locations. So, for example, a layer 2-3 projection neuron that's specific to the medial frontal anterior cortex. And then upon that, we can go further down and we can actually look at organelle structure, we can look at the neuropil, and we can actually look at individual synapses. And in fact, we can even look at synaptic vesicles at those locations. So what did we see when we used this approach? One of the first things that kind of jumped out at us was changes to mitochondria that would be classically associated with a cellular stress response. So a classic approach of looking at this is actually what's called to measure the mitochondrial aspect ratio. So basically it's an assessment of essentially how circular the mitochondria are. And essentially if they're more elongated, then they're under less stress. But what we saw in the large deletion, specifically in these layer 2-3 projection neurons, were mitochondria that would be indicative of mitochondria that are under a stress situation. And then when we looked at that into a little bit more detail, we saw some other aspects of cellular stress, classic hallmarks of cellular stress via EM. So for example, we saw a lack of invagination of the nucleus in the large deletion. Also, we saw changes in ribosome density. And also the mitochondria, I should mention, it's not just the circular nature, they're also the cristae are somewhat blown out in appearance compared to wild type layer 2-3 projection neurons. And then finally, so there's this cell stress observation, but finally when we actually looked at synaptic characteristics, we actually saw that in the dendrites of these layer 2-3 projection neurons, there was actually a reduction in synaptic density. And when we actually counted the number of synaptic vesicles at these locations, they were reduced in density also.
Was there, if I can ask, was there a,
yep.
I presume that you were always relying on tracing back to the soma, so if you had a mitochondria in 2-3, a mitochondrion, you knew it was from a 2-3 neuron, not a passing dendrite from infragranular levels.
Correct, yeah.
Right?
Yep.
So assuming you do that, did you compare to neighboring mitochondria and dendrites that were passing through that you wouldn't necessarily ascribe to layer 2-3 cells of origin?
Yes, so we definitely looked at the local neuropil, I guess you could describe it, and so in those instances, yes, you don't know the cell origin, but we did see some changes. But again, the issue is, what is the cell class that you're looking at? Go ahead.
Does the protein localize to mitochondria?
Sorry?
Does that protein localize to mitochondria?
Protein.
Of the gene, the 22q?
Yes, so there are a number of genes within the 22q11 region, but I will home in on a particular gene that we think is relevant to these observations.
So you have an increase of inhibitory neurons in this region, kind of with the decrease in excitatory neurons. Here you're looking at excitatory synapses. What happens? What's the change in inhibitory synapses on these dendritic segments or on these neurons in layer 2-3?
That's a great question. That's not something we can answer right now. I mean, we can go back and look via what you would, perhaps classic hallmarks of what an inhibitory interneuron's morphological features are via EM. It's not as clear-cut as a nice projection neuron, which has its nice, pyramidal cell structure with the apical dendrite and the basal dendrites coming off of it. So you can identify those pretty easily by EM, which don't require further kind of marker analysis. But I will come to a slide later where we're thinking of addressing those kind of questions. Okay, so again, trying to home in on particular circuit components, this layer 2-3 projection neuron circuit component in particular. So we could also then look at deeper layer projection neurons, layer 5-6 projection neurons, and compare between wild type and large deletion in the same NFAC area. And we did not see a significant difference in these various hallmarks that we saw in the large deletion layer 2-3 projection neurons. We did not see that in the 5-6.
So to push on this point, but and again, you'll probably clear it up in a minute, but I presume, again, now for the infragranular cells, you're doing the same thing. You're looking down in those layers, we can see the cell body and attribute it. But if one wanted to make an argument for a cell autonomous versus a regional effect, like up in the supragranular layers, you might want to also compare the dendrites of the deep cells, their mitochondria in layer 2.
Right.
But you can't do that with this technique, clearly, right? Unless you're lucky in plain dissection or serial reconstruction to get all the way back. Right?
Yes. Yes.
Okay.
I mean, we can do the comparable neuropil analysis that I said we did do in the layer 2-3 down in this region as well, for sure. Okay, so I kind of brought it down in resolution all the way down to the EM. Now let's blow it back up and see, are any of these changes I've described actually, could they have impinge upon the behavior of the large deletion mouse model? So what I did in collaboration with Larry Rothblatt at GW, who actually came up with this procedure, is to actually use a behavioral paradigm that is very actually known to be heavily reliant upon cortical connections. It's very heavily cortically driven and relying upon these cortical association areas. And this test is basically called a reversal learning task. So what this relies upon is the mouse is presented with a touch-sensitive screen that has 2 symbols displayed to it. And in this initial learning phase, the mouse has to learn which one of these symbols would lead to a food reward. So the wild type and the large deletion animals will do this test, this initial learning test, and I'll point out that the large deletion does not have an issue with this initial learning, so it can do that as well as the wild type animals. But when we do run into some interesting changes is the key component, the reversal component. So here, after the initial learning, suddenly the symbology gets switched, and the mouse has to learn now that it's actually the other symbol that leads to a food reward. And you can essentially quantify how long it takes for these animals to learn this switch, and it's this reversal switch that is heavily reliant upon these cognitive cortical circuits. And what we actually showed was that in the large deletion, whilst there's a range of behavioral performance, it's actually, on average, takes them significantly longer to reach this criterion by which they can say they've basically learned the reversal task. I'll point out that classic lesion, so mechanical lesion studies at sites of cortical association areas, so the MFAC, lateral entorhinal, that will actually lead to changes in the ability to perform this task, which are actually quite similar to the large deletion. So could it be that the large deletion is somewhat of a, could we say, perhaps like a genetic lesion? So you have these mechanical lesions where you're disrupting the cells. It's known from my work that the actual density of the layer 2-3 projection neurons in this MFAC region are reduced. So an argument could be made is that a subtle genetic variation of the mechanical lesions. So what we actually did was notice that there was, although the average time to perform this behavioral task would increase, there was quite a good range whereby some large deletion animals performed as well as the wild type, and clearly some did not. So what I went ahead and did was in the MFAC region of the large deletion and wild type animals, essentially assessed the neuronal density spanning that region. And what I saw was, in fact, in the animals that were poorly performing, that the layer 2-3 projection neuron density seemed to correlate with that performance ability. So that's shown here on a correlation graph, reversal sessions versus density. So, so there's that observation, and now we're trying to follow up on that with additional work. So there are ongoing studies. It's cortical association network and how it might be related to, in the large deletion, how changes are related to behavior in that relies upon that cortical association network. Yeah.
So you just mentioned you saw some variance and then that had a behavioral correlate, right? How
Right.
to these? I mean, what is the overall variance that you see in your labs?
That's a great question, and that is something that we see remarkably across, no, I don't think even us, other people who work, for example, work on the heart aspect of this, is that there certainly is variability in the penetrance of some of these phenotypes, which in some ways, makes it trickier to study. But I'll also point out in humans, you can have two humans that have exactly the same 22q11 deletion, and one might be much more severely affected than the other. So in that regard, you could say it's quite comparable to the human condition.
So have you ever actually gone and tried to cross-correlate the different assays to see if there's broad trends in terms of mice which have a lot of PNs are associated with these things?
Right. So some of the initial work, for example, the proliferation defects that I described, I mean, you're sacrificing those embryonically, so it's difficult to find a correlation there in the mature brain. But one avenue that we are interested in pursuing is, I didn't really mention this, but there is heart defects in this condition. So there's this notion that perhaps, cerebral blood flow pressure could also be affecting development. So you could potentially take animals that survive, look at their cortex, but also look at the heart at the same time and see if there's any kind of correlation there. So there's things that you can do for sure.
And in terms of the neuron phenotype that you saw, you showed this case where you have relatively normal numbers in one set of the people said relatively. Do all of the neural phenotypes kind of parse out like that? Or if you look in there, do they have an increased number of interneurons still in that population?
Right. So great question. We looked at the interneuron distribution, and that did not correlate significantly, unlike the projection neuron density. So something that we're doing at the moment is, I described these dendritic phenotypes using that GFP labeling approach. It's quite hard to do similar approaches to assess terminal fields of axons in a particular cortical region. It's not as easy as dendritic analysis coming off a cell body in that same region. One kind of way of looking at this from a different angle is, I guess I should go back a bit, is we're actually looking at the callosum in the large deletion animals. So we're sampling, again, by that block face approach where you can take large areas and then home in specifically on defined anatomical locations. And then at those defined locations, we can actually look at callosal axon bundles. So here, the callosum is essentially, you're looking at cells, layer 2 free projection neurons that are basically projecting across into the other hemisphere. This would be predominantly, mostly upper layer projection neurons that are doing this. So if you can actually assess, so these are examples, strips between a wild type and a large deletion. So, and then we can actually look at, for example, the numbers of axons, the density, the area. And so this is kind of a way of looking at the axonal projections in a little bit more detail between wild type and large deletion. And again, that's something that we could then correlate to behavior.
Are you inferring from that some sort of a clear or simple relationship between the parent axon diameter and the arborization extent, number of boutons, spread, et cetera, for a given axon? Implicit?
Yeah, I mean, it's we just started that, so I can't even really say that we have any significant observations yet. But one hypothesis is at least you would expect, at least from the stereotactic injection work, I mean, that would suggest there will be a reduced number. We'll be interested to see if there's axonal diameter differences.
I mean, how about transport processes? Any possibility your labeling is affected by how readily it retrogradely transports?
Yeah, that's possible. In which case, the SEM work would, either agree with that or not. So this is some collaborative work, preliminary work, but essentially, looking at electrophysiological responses in these cortical association areas relative to the MFAC injection site that I was talking about earlier. So basically, you can stimulate at different locations and then record the response in that MFAC region. And this is basically the methodology, an electrode that spans the cortex in that location, a multi-point electrode. And then these are some representative preliminary images, plots from that data. So for example, our collaborator has shown some differences in latency of a signal following stimulation and also intensity of signal in that MFAC region in the large deletion compared to wild types. So I'm trying to build a notion here that there are in fact changes in these cortical association networks, in the number of cells present, the arrangements, the axonal projections, the electrophysiological responses. Is there any possibility that that performance, that behavioral task can be improved upon or restored perhaps in our large deletion model? So I mentioned earlier that this observation that there were these stress observations in these large deletion layer 2-3 projection neurons in terms of mitochondrial structure and other features. So what we actually did was we administered N-acetylcysteine, NAC, which is basically an antioxidant, and we administered that to pups, and then basically redid this behavioral task with large deletion animals that had been had a NAC administered to them, and we did indeed see an improvement in this ability to perform this reversal task compared to the large deletion. And then when we looked again in more detail, going back on those previous experiments that I described to you, for example, the dendritic labeling with GFP, we saw a restoration of the apical dendrite upon administration of NAC. And then when we looked at the SEM approaches, we saw that the mitochondria looked healthier. There were more synapses, and the synaptic vesicle density had improved. And that's quantified there. The red bars in the background represent the large deletion mean plus SEM. So I mentioned that there are a number of genes within this 1.5 megabase pair deletion that are heterozygously deleted in the large deletion model. So then, we're interested to know, are there candidate genes that can perhaps explain some of this cell stress observations? So from our work and literature, it's known that there are about 6 candidate genes that have mitochondrial function within the 1.5 megabase spanning region. So we essentially did a screen, an in vitro screen, whereby we knocked down each of these mitochondrial candidate genes within in vitro cortical cultures. So the way you go about doing this is electroporating EGFP into embryonic cortex, which would EGFP co-expressing with the knockdown construct. So basically then you can dissociate those cortical cells into a culture, plate it over a certain number of days, and then you'll have neurite outgrowth that would equate to an axonal projection and dendritic arborization. And of the candidate genes that we screened across, we found that TXNRD2 actually reduced this neurite outgrowth relative to control knockdown experiments. And I'll point out that these observations replicated what we saw when we basically plated large deletion cortical cultures. So the large deletion cortical culture grown under the same paradigm looked comparable to the TXNRD2 knockdown. And that's the quantified results there. So TXNRD2 is part of the thioreductase system, which is basically an endogenous antioxidant defense system within cells. So essentially they reduce oxidized amino acids and also reactive oxygen species. So TXNRD2, when we actually looked at using basically mitosocs, which is a fluorescent reporter that basically the fluorescence is related to the amount of reactive oxygen species within those cortical cultures, we could actually look at the intensity of the mitosoc signal and the TXNRD2 knockdowns versus control experiments. And we saw that mitochondrial ROS was elevated in the TXNRD2 knockdowns, which would make sense based off its known role. And then when we look in vivo, when we look in tissue sections, so this is basically the cortex from upper layer to lower layers with a cortical neuronal marker there. We actually show that TXNRD2 in red was quite relatively abundantly expressed in those upper layer 2-3 projection neurons. And then looking at a TXNRD2 het line, so this is and specifically again using that CUX2 line, so you can actually heterozygously delete one copy of TXNRD2 specifically in these layer 2-3 projection neurons. So they're expressing 50% of TXNRD2. What we actually saw was that there was a reduction in this line in the apical dendritic complexity compared to wild types. So that would be comparable to what we saw in the large deletion line. So this is pretty new preliminary data, but I threw it in here. This is the reversal task, but again, now this is with not the large deletion line, but this is in fact a constitutive TXNRD2 het. So all cells are heterozygously expressing TXNRD2 in this animal. When we perform this reversal task, it does actually show with what is a more limited data set, because this is preliminary, but nevertheless it does show that it takes these animals longer to perform this task than the wild types. Remains to be seen if it will be somewhat intermediate between the large deletion and the wild type. That could possibly be the case. So where it actually diverges from the large deletion phenotype is quite interesting to me. So again, this is preliminary, but we're getting decent numbers. So this is assessing the upper layer projection neuron density in the MFAC region, and we don't actually so far see a significant difference between the wild type and the TXNRD2. So I said that there is a difference, nevertheless, in the large deletion. So here we're coming into an issue of, could it be perhaps a combination of factors that are actually at play in the large deletion in terms of that behavioral response? So you have perhaps the local microcircuitry in terms of dendritic complexity and the cell stress of those layer 2-3 projection neurons. Then you also have these embryonic kind of factors feeding into it in terms of reduced layer 2-3 projection neurons. So we can formally test some of that in more detail by essentially continuing what we've started to do in the large deletion, whereby we can assess this exonal density, number, diameter in the TXNRD2 line. And we can see, for example, if for example, there is not a reduction there, then that would suggest that any of the behavioral response we're seeing is not necessarily critically dependent upon these long-range exonal projections between the association areas. And we can also do some of these electrophys experiments as well. Another thing that we're gearing up to do, and comes to your point, I think, that you mentioned earlier, is you can use these kind of somewhat new approaches whereby you can actually, under cell-specific genetic control, you can actually express EM contrasting agents in specific cell types. So here you could envisage that perhaps, when I was talking about the synaptic number and the synaptic vesicles that are actually forming synapses onto these large layer 2-3 projection neurons, we can actually get a sense of what the cell types are that are forming these synapses with the 2-3 projection neurons. So to try and bring that all together, I've indicated that there are these connection issues in the large deletion as shown via our stereotactic injection work. And in large deletion, there are dendritic morphology changes. Those morphology changes can be replicated when we use the TXNRD2 in vitro and in vivo work. And so then we've also now shown that cognitive behavior in the TXNRD2 is also perturbed. So could there be a compounding effect here that's kind of being revealed by the TXNRD2 that occurs over and above the embryonic cortical events that perhaps are not TXNRD2 dependent. And if I basically summarize that all together in one big slide, we have these embryonic cortical interneurons that move into the large deletion cortex that are altered in their location, laminar location. We have the proliferation defect for the large deletion layer 2-3 projection neurons. That seems to result in reduced numbers of axonal projections between the cortical association areas. And then we have this locally disrupted dendrites and synapses that would seem to have a TXNRD2 component. With that, I'll be happy to take any questions.
Have you tried giving the TXNRD2 back to the large deletion mouse?
In vitro, we have. So the you remember the neurite complexity in the large deletion. If we overexpress TXNRD2 in vitro in that cortical assay system, we do see an improvement in the dendritic complexity in vitro.
The interneurons that are there, so typically in development, those GABergic cells, because of the change in the chloride equilibrium potential, are excitatory early on. And so while we tend to think about in the established adult circuit the kind of excitatory inhibitory balance difference you show with those cells, it very well could be that that circuit is eleptogenic early on in development. So I see you're doing some of the electrophysiology. Do you have any sense of that this time?
That electrophysics work is in collaboration, so it's not really my field, but it is interesting. For example, maybe there's a latency in that switch from excitatory to inhibitory. Maybe that timing of that is off in the large deletion. That's something that we thought about and I think could be worth looking at.
It's something like that has certainly been implied in other animal models of autism and so forth. Exactly that. That's everything about that. So. Yep.
My question kind of follows up with that in terms of what is it about the layer 2-3 neurons early on where you do see that you saw the mitochondrial defect early, right? And that might have an effect in the later populations of migration. And then you also still see the mitochondrial defect in the adult. So is there an energy demand difference early on, right? The lateral around the cortex is going to come on until you have experience-dependent plasticity. So can you speak a little bit more?
Right. So the EM work, that was all mature cortex. But and I'll also point out, just from expression analyses we've done in the past, TXNRD2 is minimally expressed embryonically. It kind of switches on, kind of really ramps up actually at P0 onwards. So in that's another, the lack of a difference in terms of projection neuron density in the TXNRD2 het, that would jibe with that because it's probably not got an embryonic presence. So, I'm leaning towards at least birth onwards for perhaps cell stress events that we're seeing in the neurons. That could be TXNRD2 dependent. Now, I did point out there was 6 candidate genes, so we can't formally exclude that those other guys might be having a role embryonically.
But so, like, in contrast to layer 5, 6, where you don't see mitochondrial, because is there something, maybe you stress the animals where now layer 5, 6 need to engage the mitochondria, would you induce the effect, and there's something about layer 2, 3 that are predisposing them to the mitochondrial defect?
Yes, that would be an experiment that we could do, so we could certainly stress the animals. Our current thinking is that certainly in the MFAC, it's this kind of a crazy region that integrates a lot of crosstalk between these cortical association areas that would be predominantly mediated through layer 2, 3. So those cells might be particularly active.
So you showed us that there were like 6 mitochondrial genes on the Q, and you have narrowed it down to 1. So I was wondering if you have looked at effects of others and potential possibility of what they do.
Of other what, sorry?
There were like 6
Yes.
genes. Have you looked at the other 5 and the potential possibility of their role in the phenotype?
Right, so the in vitro screen would suggest that in the the neurite outgrowth, at least, that they're not the key players, unlike TXNRD2. But as we were discussing, I mean, for example, the embryonic genesis of projection neurons, the migration of cortical interneurons, they're heavily energy-dependent processes, presumably, so I can't formally exclude that those other genes are involved in that, at least.
One of them was the RTM4R. That's an axon growth molecule. I don't know much about it besides that.
It's, yeah, I mean, I know it resides in the region. There's not much literature on it, to be honest, relevant to this. And also, I think in previous work that I think Tom Maynard's done, he's actually had difficulty trying to actually pick it up in some of these embryonic cortical structures, at least. So it's not something that we've really focused on.
Yeah, so the proliferation that you showed at the beginning of the study, you showed only one time point, I guess, and sometimes in autism what happens that if you have like a delayed proliferation, have a change in that, so did you check different time points to see if later you can have like some compensation, or if it's only just a change in development and not only every time?
Right. So we are actually, although that paper's quite old, we have actually revisited it because I mean, the observations still stand, but some of the field in proliferation has kind of moved on a bit since then. So what we're actually trying to figure out is, for example, that basal progenitor pool that I mentioned. You know, one issue that we're trying to figure out is, so that pool is supposed to only give rise to maybe 1 to 2 to 3 divisions before it's done, basically. So we're trying to kind of home in on that a little bit more using various co-markers for neurons and newly formed neurons. So basically within the cortex, you can kind of assess in that basal progenitor pool what is the kind of terminal fraction, the terminally dividing fraction of cells that are immediately giving rise to neurons. Excuse me. So we're trying to see if there's a difference in the kinetics of that in the wild type versus the large deletion.
So I thought one of the most interesting parts is that you're getting these PD cells that are supposed to be in layer 5 migrating up into layer 2, 3. And so what happens in layer 5 in terms of the circuitry? And one would expect these mice to have seizures probably because of a loss of the PD inputs if the PD inputs aren't in there.
Right. I mean, go ahead.
Those synapses and cells are really important for critical period plasticity and all these other things. So what happens in these mice in those sorts of assays?
Well, two things. I mean, there is a shift, but it's a quantitative shift. So we're not talking like a massive, you know, I probably should have actually shown the actual images instead of just a cartoon, but it's more of a kind of a quantitative shift. So we're not seeing like a complete removal of cells from one layer to the next. In human literature, epilepsy is, if I'm remembering correctly, in the 22q11 cohort is elevated against the general population. So, you know, we have thought that could, you know, the logical conclusion would be that could be something down to the parvalbumin distribution that I showed had changed. But yeah, it's all the things in the large deletion as in the human case. It's kind of like a subtle shift. So there's variability. You know, one animal might show more pronounced change in the parvalbumin distribution than the other. So as I mentioned, it kind of makes it hard to study, but it also seemingly is what's going on in the human condition.
But to your point then, is it, I realize you showed us kind of a summary cartoon of that in the layers. So is it that you're sure that those cells have migrated, or are there more of them overall, or of true quantitative redistribution?
Right. So yes. So we did. I mean, we just counted them. So we basically assessed the density, and there wasn't that there was a reduction or an increase. It was actually a quantitative shift in their locations. And interestingly, looking at a couple of different areas within the cortex, where you are in the cortex, those shifts are not always the same. So there is a shift, but it's not necessarily exactly the same as what I showed in that cartoon slide for simplicity's sake. But,
cells are really the last to be incorporated of the interneurons into functional circuits, and so maybe they're the most likely to be able to move up.
Right. So I didn't show it, but I did live imaging basically of the cortical interneurons going into the cortex embryonically. But as you point out, actually there's a lot of radial distribution in the first postnatal week of those cortical interneurons, and that's a lot harder to live image. But it's quite possible that something is going on there that we don't have an exact handle on. But yeah, what was your sorry? What was the second?
Whether, because those PB cells are so important for kind of closing the critical period in a lot of parts of the cortex, do these animals have any sort of defects in kind of the deprivation?
Yeah, that is one area that's understudied, I think, in this model, to be honest with you.
Let me go to that one. Okay.
How much of the overall human phenotype do you get in your large deletion?
Right, so that's dependent upon the structure that you're looking at. So for example, the heart phenotype, it's a very characteristic third arch phenotype during the development of the heart. And that's seen in humans, and quite remarkably, the very similar third arch phenotype is seen in the mice. So that's like a nice kind of confirmation that I guess its underlying biology is comparable between the mouse and the humans. Obviously, when you start moving into the brain and behaviors, then you run into the usual issues of mouse versus human. But we tried to use experimental paradigms that we think mimic cortical activity, cortical association areas as best we could. Yes, so Tom Maynard, I think actually when he was here, he might have talked a bit about how there are some subtle craniofacial changes in the large deletion, that you could argue are comparable to the human condition.
I'm going to change my question because I'm interested in what you just said. Do you see the mitochondrial? Do you see the mitochondrial phenotype in the heart? Is this like have you checked like the liver, something that relies on fatty acid? Is this like an energetic deficit that could be?
Great question, but no, we haven't done that. But
Okay, my second question, which was, I was really interested that you showed that there's a decrease in ribosome density also in your EM data. Was that associated with mitochondria, suggesting like the loss of M, the mitochondrial ribosomal protein 40 that's lost in this deletion? Is it mitochondrial translation that's disrupted, or is it general ribosome? Is it throughout?
yeah, that was more of an observation that wasn't quantified, but you can actually quantify that if find a undergrad or a grad student who wants to count those little rosettes. But
So they were rosettes, though, so that was not.
they were rosettes,
Okay,
yes.
Okay, why don't we call it and thank Dan again for his seminar. Appreciate it.